In this video, we're going to look at methods we use in the lab that rely on nucleic acid hybridisation. You need to learn the theory behind the tools of the trade that you may use one day in the lab. All nucleic acid hybridisation is the joining of two DNA or RNA strands via their base pairs. And in the lab, we can use this base pairing characteristic in our experiments. A lot of the methods in the lab that rely on nucleic acid hybridisation have the same characteristics. That is, we have a target piece of DNA or RNA in a mixed sample and another piece of RNA or DNA that is a probe and is coupled to a label that is usually radioactive or fluorescent. We then bind the sample to the solid support, which is often referred to as a membrane, and if our sample is DNA, we then have to denature it, that is, break the current base pairing with heat or alkali, and then we use the probe to see if our target is present. The idea here is that the base sequence of the probe will match and bind to the target in the DNA or RNA on the membrane. The polymerase chain reaction works in a similar way and relies on DNA hybridisation events, except in PCR there is no solid support and the probes are primers and the signal is not radioactive or fluorescent, but is the production of millions of copies of the target DNA. Now, when we are doing this type of experiment, there are two critical parameters that we need to consider. First is specificity. That is, will the probe only stick to one target sequence or multiple targets? And second is sensitivity. That is, will we need a lot of the probe to detect the sequence in the other molecule? We can improve specificity by designing the probes so that they only match the target of interest and have a melting temperature that is ideal for the experiment. And through the use of sodium chloride and temperature, we can get stringency. That is, we can control how well the bases have to match for the probe to bind and for us to get a signal. For example, if we have a high salt concentration and a low temperature, we get low stringency. That is, we get poor matching of bases, so we may get a lot of false positives. On the other hand, if we have low salt concentrations and a high temperature, then we get high stringency. And that means that we have to have a very good match between the bases to get the probe to stick. However, this may lead to false negatives. Hence, through the use of salt and temperature, we can control the specificity of binding. So, what are some lab methods that rely on DNA hybridisation? Well, we have PCR. We have dot-blot hybridisation, which has a DNA target in an oligonucleotide probe. Dot-blot hybridisation is a very simple method in which the sample is spotted onto a support material, a membrane, and then dried and then probed with the target DNA. We have southern blot hybridisation. This has a DNA target and a DNA probe. This method was first published by British biologist Sir Edwin Southern and represents the start of a joke. Next, there is northern blot hybridisation, which uses RNA as the target and DNA as the probe. And this is the joke. The first blotting method described was named after a person, Southern, but the following blotting methods, such as Northern, were named after points on the compass. There is also a protein blotting method that uses antibodies as the probes and is called Western blotting. Anyway, back to the methods. Colony blot hybridisation, which has a bacterial DNA target and a DNA probe. In this method, an impression of a bacterial plate is taken, the DNA fixed to a sheet of membrane and then probed. This method allows you to identify bacteria that contain or have taken up a particular piece of DNA. Next, chromosome in situ hybridisation. The whole chromosome is the target here, and the DNA is used as a probe to paint the chromosome to show the location of specific sequences and genes. Gene expression analysis, also known as microarrays. Here we have a cDNA as a target on a chip, and then we probe with messenger RNAs from the cell. This method is used for detecting which genes are being expressed in a population of cells. And finally, we have gene editing using the CRISPR-Cas9 system, which is a method that allows targeted editing of genes.